This lesson will introduce non-clinical safety evaluation for in vivo gene therapy products. In the development of gene therapy products, several non-clinical safety evaluations are required to support clinical trials. According to the Guideline on Ensuring the Quality and Safety of Gene Therapy Products, there are 6 points to consider. Vector property, general toxicity, including safety pharmacology, gene integration, carcinogenicity, reproductive and developmental toxicity, and immunotoxicity. Understanding vector properties. First of all, it is necessary to understand the functional properties of the promoter and transgene. Second, in the case of a viral vector, the properties of the wild-type virus from which it is derived are informative. There are several important points to understand. Physicochemical stability, pathogenicity, cytotoxicity, species specificity, tissue or cell tropism, transduction or expression efficiency, mechanism of integration into chromosomes, and replication properties of the vector. This information helps to select animal species, interpret study results in the general toxicity study, and estimate the risk of carcinogenicity and reproductive toxicity. General toxicity study design. It is important to use pharmacologically relevant animals and the same route of administration as in the clinical trials. Selection of high dose for hazard evaluation and determination of a sufficient observation period for toxicological evaluation are also important. For the observations, endpoints should include clinical observation, body weight, food consumption, complete blood count, serum chemistry, urinalysis, ophthalmology, organ weights, and gross and microscopic pathology of organs or tissues with reference to the endpoints in the general toxicity studies of pharmaceuticals. However, the study design can be set on a case-by-case basis, based on the properties of the product and existing information. Gene integration evaluation. The risk of unintended gene integration into chromosomes should be evaluated because gene integration may lead to an increase in carcinogenic potential or transmission of vector DNA to progeny. Risk of gene integration should be assessed by the number of viral copies per cell, or integration site analysis, considering some points like integration machinery, cell differentiation, dose and route of administration. In addition, evaluation of the vector distribution in reproductive organs and its changes is required to estimate the risk of integration into germ cells. Other points to consider. This slide explains carcinogenicity, reproductive and developmental toxicity, and immunotoxicity. First, carcinogenicity. It should be evaluated in consideration of the results of gene integration analysis, results of a general toxicity study, and the biological properties of the expressed protein. Standard rodent carcinogenicity studies used for chemical drugs are not appropriate for gene therapy products because possible mechanisms of carcinogenesis attributable to gene therapy products include gene integration into chromosomes and biological properties of the expressed protein. Second, the reproductive and developmental toxicity. Effects on reproductive and developmental toxicity should be assessed by the result of biodistribution and vector properties. In addition, effects on fertility are evaluated by histopathological examinations of reproductive organs in the general toxicity studies. The need for reproductive and developmental toxicity study should be considered from the information of the intended patient population and the biological properties of the products. Lastly, the immunotoxicity. The risk of immunotoxicity, such as producing anti-vector antibodies, is evaluated in general toxicity studies. But the study results should be interpreted in light of the potential of inducing specific immune responses in animal studies. Conclusion. There are several basic points on non-clinical safety evaluation of gene therapy products. Understanding the properties of the vector. Selection of the animal species and interpretation of the results. Collecting a wide range of existing information on risks, such as literature on use results of similar products in humans, and taking appropriate risk mitigation measures in clinical trials. As science and technology advance, various types of gene therapy products are developed. Therefore, a case-by-case safety evaluation based on the unique properties of each product is necessary.